Our understanding of how the Lac Operon is regulated in response to the availability of lactose in the environment is because of pioneering experiments by Jacob and Monod, for which they won the Nobel Prize. And let's discuss the kind of genetic experiments they did that allowed us to understand how the Lac Operon functions. So they used a number of mutants, and let's write down both the wild type as well as the mutant alleles of these genes. The first one is Lac Z minus, or Z minus for short, where beta gal is not functional, and this E. coli strain, which is Z minus, cannot grow in medium with lactose, because if you don't have functional beta gal, then you cannot metabolize lactose. They also had Lac Y minus, which would have a similar phenotype and an inability to grow in a medium with lactose as the only sugar, because in the absence of the functional permease, you would not be able to transport lactose into the cell. A third type of mutant they had was of the operator OC, and the wild type is written as O plus, and the OC strain of E. coli has mutant operator sequence. And the final class of mutants are for Lac in the Lac I gene that makes a repressor. There are two of them. There's I minus and IS, where the wild type is written as I plus. A second thing worth mentioning is, along with these mutant strains, how did Jacob and Monod induce Lac Operon expression? And if you recall, lactose gets converted to allolactose, which acts as an allosteric inducer of the Lac repressor, by which we mean that allolactose binds to the Lac repressor and causes changes in its activity, in its ability to bind the operator DNA, as a result of conformational or shape changes in the protein that result from allolactose's binding to the repressor. The challenge with using lactose as the inducer, although in nature lactose is the inducer, but the challenge with using lactose as an inducer in an experimental setting is that once the Lac Operon is induced, its transcription is induced, and beta gal protein is made, beta gal will then break lactose down into galactose and glucose, and the amount or the concentration of lactose will change during the course of the experiment, making the interpretation of the experiment hard. So instead of using lactose as an inducer, Jacob and Monod use isopropyl beta D-thiogalactoside IPTG as the inducer, and this compound is structurally similar to allolactose, and therefore it can act as an inducer, as an allosteric inducer of the Lac repressor. However, it is not a substrate for beta gal, and therefore its concentration or amount stays constant during the course of the experiment. So the kind of experiment that Jacob and Monod performed was that they would take strains of a particular genotype of the Lac Operon and then grow these strains either in the absence or without IPTG, which is the same thing as growing them without lactose, or in the presence of IPTG, and then measure or assay whether beta gal and permease are expressed or induced or not. So let's do the wild type case, and this is fairly straightforward. We have I plus, which is lac-I, which makes the repressor. Then we have the promoter, and after that we have the operator, and in this case the operator is O plus. Then we have Z plus and Y plus, and we know that when you don't have lactose, lac-I is going to make the repressor, which is going to go and bind to the operator and prevent the polymerase from elongating. And therefore, when you don't have lactose, or that is in the minus IPTG condition, neither Z nor Y will be expressed, and so we can say that we will have minuses over here. On the other hand, when we have lactose available in the cell, then the repressor will still be made, will be still be transcribed and translated from the lac-I gene. However, this repressor is going to change shape because IPTG is going to bind to it, and as a result, this allosterically induced repressor molecule will not be able to bind the operator sequence, and the polymerase will be able to bind and transcribe, and therefore both Z and Y will be expressed. So we can say plus in these two columns. And this, if you notice the pattern, that when you don't have IPTG, then you don't have expression. So there's a minus, whereas when you have IPTG or when you have lactose, you induce expression. And so if you see a pattern of minus plus, we call this inducible. Next, let us look at the next genotype where the operator and Y are wild type, but we have a Z minus mutant. The function of the repressor will be exactly the same as before, with the difference that our genotypes are a little bit different. We have O plus, Z minus Y plus, and O plus, Z minus Y plus. Now, when we are looking at the no lactose condition, since the repressor is bound at the operator and no transcription happens, you don't express any of the genes, neither Z nor Y, and therefore we will detect neither beta gal nor permease. However, when lactose is available to the cell, because of allosteric induction, the repressor will not be able to bind to the operator sequence, and the polymerase will elongate. With the difference that since we have a mutant allele of Lac Z, Z minus, we will not get functional beta gal, and therefore you will have a minus over here and a plus for permease since we have a Y plus allele. In the next genotype, instead of having a mutant Z allele, we have a mutant Y allele, and therefore we have Z plus and Y minus. And the status of whether repressor is bound at the operator and polymerase is elongating or not does not change. The only difference is that when you don't have lactose, so in the minus IPTG condition, we still don't get expression of either beta gal or permease, since the repressor prevents the polymerase from binding the promoter and transcribing. But in the condition where we have lactose in the cell, polymerase can elongate because the repressor can no longer bind the operator due to allosteric induction. However, we will get a functional beta gal protein since we have a Z plus allele, but a non-functional permease protein, and therefore we will get a plus over here and a minus over here. And it's important to keep in mind that we are using our understanding of the Lac Operon to determine what these phenotypes are. However, when Jacob and Monod did these experiments, they were working in reverse, where they inferred how the Lac Operon works by looking at the results of these experiments. Next, Jacob and Monod investigated whether the Z minus, the mutant allele of Lac Z, was dominant or recessive. Normally, since bacteria are haploid organisms, we can't talk about dominance or recessiveness, but recall that you can use partial diploids in bacteria to create strains that are diploid for certain genes or certain sections of the chromosome. And the way you do that is that, let's say we have an F prime strain, which means that the cells have a bacterial chromosome, but they also have a plasmid, an F plasmid, that carries a section of the bacterial chromosome on it. And that's because this cell was derived from an HFR cell and the plasmid popped out, but when it popped out, it took a section of the bacterial chromosome with itself. And furthermore, let's say this F prime strain is Z minus, which means it carries the F prime plasmid carries with it the Z minus allele from the bacterial chromosome. And now let's mate this F prime cell with an F minus cell that is Z plus, which means that this F minus cell has its chromosome, and on that chromosome it has the wild type allele of the LAC-Z gene. So when these two cells conjugate, through rolling circle replication, the F prime plasmid is going to get transferred to the F minus cell, and on that F prime plasmid will be the Z minus allele, and we would have created a bacterial strain that's diploid for this section of the chromosome, in other words, a partial diploid, and we would write its genotype as F minus Z plus and over F prime Z minus. So Jacob and Monod used partial diploid strains. They constructed partial diploid strains that contained different alleles of the same gene, and were therefore able to study the dominance relationships between these alleles. And we should keep in mind that when we are looking at such genotypes, we are looking at cells that have two pieces of DNA in them. One piece of DNA is the bacterial chromosome, whose genotype is written like this, and the other piece of DNA is the F prime plasmid, whose genotype is written in this manner. And the first partial diploid that Jacob and Monod investigated was O plus Z plus Y plus. So this was an F minus strain, which was a wild type, and then this was mated with an F prime strain with the genotype O plus Z minus Y plus, giving us a partial diploid where you are a heterozygote for the two alleles of LAC-Z. When Jacob and Monod looked at the expression of beta-gal and permease under the two conditions, without lactose and with lactose, what they observed was that the beta-gal was inducible. Now recall that Z minus leads to no beta-gal expression, but in a partial diploid, with a heterozygote with a Z plus allele, the expression of beta-gal becomes inducible again. It changes when you add lactose, so it changes from minus to plus, and therefore if you have a diploid between these two genotypes, the phenotype of the Z plus allele wins out, and therefore we would conclude that Z plus is dominant over Z minus. And it's worthwhile to look at this in a mechanistic manner. So when you have no lactose, the repressor protein binds to the operator on both the DNA molecules on the bacterial chromosome as well as the F prime plasmid, preventing transcription, and therefore you get no expression. So neither beta gal nor permease is expressed from either allele. On the other hand, when you have lactose in the cell, and let me quickly write the genotypes down. You will allosterically induce the repressor, so the repressor is going to have a different shape. Because of binding by IPTG, which will then not be able to bind the operator site on either DNA molecule, and therefore we will have transcription on both the DNA molecules, and we will get beta gal protein expressed as well as permease protein expressed, since one of the alleles is Z plus, and that's why we see both beta gal and permease expressed. And of course this assumes that beta gal is is haplosufficient. Nevertheless, the result of this experiment, which is the winning out of the Z plus phenotype over the Z minus phenotype in the partial diploid, led to the conclusion that Z plus is dominant over Z minus. Next, let us look at Jacobian monods results when they tested the mutant for the operator site OC. When they looked at E. coli strains that have the OC allele, the mutant allele of the operator site, they found that both beta gal and permease were expressed even in the absence of IPTG. And in other words, beta gal and permease were expressed irrespective of the condition or in all conditions, and this is called as constitutive expression or constitutive mutant. And let us see how this might work mechanistically based on our understanding of the Lac Operon. So we have I plus, OC, Z plus, and Y plus. And the Lac I gene makes the repressor, which normally would go and bind the operator site. And let's do the condition that we don't have any lactose first. However, if the mutations in the operator site changed its DNA sequence such that the repressor no longer recognizes the operator site and does not bind there, that would imply that the polymerase can bind now and carry out transcription, resulting in expression of both beta gal and permease even in the absence of IPTG or even in the absence of lactose. And if you do have lactose available in the cell, then as before, this repressor is going to bind to IPTG or allolactose, change shape, and not be able to bind the operator site. And in this case, there are two reasons why the allosterically induced repressor can't bind the operator. One, even if the operator site were the wild type sequence, the repressor whose shape has changed after IPTG or allolactose binds to it is unable to bind. And the second reason is that the sequence of OC has also changed. Therefore, polymerase will bind and carry out transcription, leading to expression of both beta-gal and permease. So this model that the OC mutant has a DNA sequence so that the repressor can't recognize it or bind to it leads to the prediction of constitutive expression, and that's how Jacob and Monod inferred or deduced that the repressor must be binding to the operator site. Next, Jacob and Monod considered a partial diploid in which the O plus allele was paired with the Y plus allele, and the OC, the mutant operator allele, was paired with a Z plus. When they performed this experiment, the result was that Y, which was paired with a wild type operator allele, was inducible as before. So it matched the wild type. However, Z, which was paired with the constitutive mutant OC for the operator, became constitutive. And recall that this is one piece of DNA and this is another piece of DNA. This implies that the operator only affects those alleles that are on the same piece of DNA as the mutant allele of the operator. And another way to put it is the operator acts on the same side, which is written as the operator acts in cis. And so on same side, and this should be read as on same DNA. And now let's see how this might work mechanistically. So I have I plus, and in the first piece of DNA I have O plus Z minus Y plus, whereas in the second piece of DNA on the F prime plasmid I have OC Z plus Y minus. Now the I plus allele is going to make a functional repressor And this repressor will go and bind the operator on the bacterial chromosome, but not on the F plasmid. And what that means is that the polymerase will not be able to elongate on the bacterial chromosome, which has the Y plus allele, and therefore Y, the permease, is going to be minus in the absence of IPTG. The Z plus allele, however, lies on the same piece of DNA on the F prime plasmid, and here, since we have an OC mutant, the repressor is not able to bind and polymerase can elongate, and we will get expression of functional beta gal in the absence of IPTG or lactose. Now, when we do have lactose, then the repressor is going to change shape because of allosteric induction, and then the repressor will not be able to bind the operator. And here it doesn't matter if you have a wild type operator allele or a mutant allele, either way the repressor is unable to bind, and therefore the polymerase will elongate on both the bacterial chromosome as well as the F prime plasmid, leading to expression of both permease as well as beta gal. The end result is that beta gal has constitutive expression matching the phenotype of the OC mutant, and the permease will have an inducible phenotype matching that of the O plus allele. And the end result, or the final conclusion from this experiment, is that the operator acts in cis, and we can see how from the structure of the Lac Operon, the fact that the operator sits between the promoter and Lac Z and Lac Y and so on matches up or is able to explain the result of this experiment. And that's how Jacob and Monod in fact figured out that the operator must be the DNA sequence that is on the same molecule, same DNA molecule as the Lac Z gene and the Lac Y gene.